One of the really cool things about prokaryotes is their wide variety of ways to get energy from their environment. So oftentimes we will categorize prokaryotes based on how they get energy from their environment. So that's what we're going to talk about here. The massive diversity of metabolic processes that prokaryotes can use. So we're going to talk about a lot of terminology here, and we are going to use some prefixes and suffixes for words that will be useful here, that will be useful in other classes that you continue on to take, and they'll be useful in 212 and 213. So I want you to focus less on memorizing the definitions of each of these words and more on trying to understand the different components of the word and what they mean. All life on a planet can obtain matter, organic matter, from two sources, either autotrophy or heterotrophy. We need molecules, we need organic matter to continue to sustain ourselves, to replace dead organelles, to copy our DNA, right? To replace our damaged membranes. So we need an input of matter. And where we get that matter, where we get that organic matter, differs based on our evolutionary history, right? But we use this term troph. You'll see this suffix troph, and that just means nourishment. So that really just means where are we getting our matter. Organisms can be autotrophs, meaning that they self-feed, okay? Or they can be heterotrophs. Hetero meaning that they get their organic matter from a different source. Okay, so again, auto means self, so self-feeding. So these are organisms that can often build their own organic compounds from inorganic substances. Heterotrophs eat other organisms, eat other organic compounds to nourish their own organic matter needs. Hetero, like us, we have to eat other organisms to get our organic matter. Autotrophs feed themselves. They produce their own organic compounds. Organisms can also differ in where do they get their energy. Where does that energy come from? Some organisms can use light, solar energy, to build their organic compounds and to carry out life processes. Other organisms use chemicals to get their energy and to fuel life's processes. Okay, so we're going to add on some additional prefixes here to see where does an organism get its energy, right? So we talked about where it gets its matter, but where do organisms get its energy from? What source? If that organism gets its energy by breaking down other molecules, we call it a chemotroph. So chemoheterotroph produces its own energy by breaking down molecules from other organisms or food that it eats. Chemoautotroph builds its own food, but then breaks it down. Or organisms can use light, right? Photo is light. Photo is the root word for light. So that gives us photoheterotrophs. So they get their energy from the sunlight, but yet they get their organic molecules or organic matter from eating something else. Or photoautotrophs, organisms that use the solar energy, use the sunlight to build their own organic molecules. Okay, let's focus for a second just on the heterotrophs, the different types of heterotrophs that we have. We have chemoheterotrophs and we have photoheterotrophs. So both of these groups are getting their carbon from external sources, right? They're eating other organisms, they're getting their carbon compounds from their food. So chemoheterotrophs are like us. We eat our food, it contains organic compounds, and then we use those organic compounds, we break them down also to get energy, right? So all animals are chemoheterotrophs. Most prokaryotes are also going to be chemoheterotrophs as well. This kind of weird group of photoheterotrophs, they still get their carbon energy from their food that they eat, from their environment, but they get their energy from sunlight. So only a very small portion of prokaryotes would be photoheterotrophs. This is kind of a rare group. There's not a whole lot that fall into this group. So they're still eating, they're still getting their carbon from their environment, but their energy that they get comes from sunlight. Okay, so some halophiles, so organisms that live in very salty environments, so halo means salt and phile means to love, would be photoheterotrophs. And again, we only see these in prokaryotes, only a very small portion of prokaryotes. So their energy comes from the sun, but their carbon, their organic matter, comes from the food that they eat. All right, now again, types of autotrophs. So these are organisms that build their own organic molecules, usually from inorganic sources. So we are most familiar with photoautotrophs. Photoautotrophs are things like plants, anything that can photosynthesize to be a photoautotroph, where their energy comes from sunlight, and the carbon comes from an inorganic source. So they can build their own organic molecules, and the energy that they use to power their systems comes from sunlight. Okay, so photoautotrophs are the most common. So plants, some protists, and cyanobacteria would be part of this group of photoautotrophs. These chemoautotrophs, though, this is kind of a weird group. They still get their carbon from carbon dioxide, but their energy that they get to kind of power their cells processes as well as convert that CO2 into an organic molecule comes from breaking down inorganic molecules. So these could be things like breaking down hydrogen sulfide or ammonia in their environment. So that process of breaking down those molecules is what gives them energy to form their organic molecules. So photo uses sunlight, chemoautotrophs use chemical energy from the environment, hydrogen sulfide, ammonia, the most common, but they're getting their carbon from an inorganic source, from carbon dioxide, not by eating other organisms. So these are only prokaryotes. We typically find these types of organisms in deep sea hydrothermal vents, where things like ammonia and hydrogen sulfide are very common. And so it's a readily available energy source that they can use to build organic compounds. So oxygen wasn't always super common in our atmosphere. Okay, there was for lots of Earth's history, especially in early Earth's history where life was around, when life started, there wasn't a lot of oxygen around. And so we see some prokaryotes that differ in their oxygen requirements or in the levels of oxygen that they can tolerate. So AER stands basically just for oxygen. And so we have some prokaryotes are anaerobic, meaning that they prefer low oxygen environments, and some prokaryotes are aerobic. They can function in oxygen-rich environments. And the reason why oxygen is important is because oxygen is what basically helps us finish off the process of cellular respiration, the process that we use to build ATP. And so with oxygen around, we can build a lot of ATP through the process of aerobic respiration, right? But not all prokaryotes can do this. Not all prokaryotes need that oxygen. And in fact, oxygen can be damaging to some organisms if they aren't used to functioning in that environment. So oxygen is helpful if organisms can go through full cellular respiration to make lots of ATP, but it can be harmful if that organism doesn't have those cellular processes in place. Because oxygen is really greedy with electrons, it'll sometimes strip other molecules of electrons. So we have some organisms that are aerobic prokaryotes, and they can go through cellular respiration in the presence of oxygen. But most prokaryotes are going to be anaerobic prokaryotes. They get their energy, their ATP molecule, from going through a process of fermentation, which is a process of anaerobic respiration, gathering energy and building ATP without the presence of oxygen, without going through the full process of cellular respiration, aerobic cellular respiration, right? We're probably familiar with anaerobic respiration or fermentation. If you've made bread before, right? Yeast goes through anaerobic fermentation. This is how we get things like beer and wine, alcohol, things like that, through anaerobic fermentation. So most prokaryotes are going to fall into here, this anaerobic preference for their lifestyle. Don't worry about the chemical equations for how fermentation happens and how much ATP is produced. That's something we'll tackle in 212. We'll tackle that later. But know that most prokaryotes are anaerobic. One of the most amazing things, I think, about prokaryotes is that they are some of the only organisms that can fix nitrogen gas. So that term fix basically means to turn something from inorganic to organic. So nitrogen gas is all around us in our atmosphere, okay? It's like 78% of our atmosphere is nitrogen gas, like this. Two nitrogens combined together with a triple bond between them. Most organisms, we need nitrogen. It's a huge component of our DNA and our RNA, and it's just, we need a lot of nitrogen. But not many organisms can break up that triple bond. That's a really strong bond, yeah? But some prokaryotes, like we see these little root nodules right here on the roots of this plant, these are prokaryotes that can turn this gaseous nitrogen with that triple bond into ammonia. And ammonia is a usable form of nitrogen. Living organisms can use this and then recombine this ammonia to build nucleic acids, to build proteins and amino acids, things like that. Nitrogen gas, not accessible to living organisms. Ammonia, some organisms can do this. But really only prokaryotes are the most common organism that can break up that nitrogen gas into ammonia and fix that nitrogen. Lastly, even though prokaryotes have a wide diversity of metabolic processes, it's important to know that they often work together and create communities that we call biofilms. So biofilms are groups of microbes that kind of work together. They sometimes create a sugary substance to kind of stick them together to create these films. So biofilms can be helpful or harmful to humans. So like plaque, the reason why we have to brush and floss our teeth, is a biofilm, a bacteria that can secrete acid that decays our teeth, right? We can also use biofilms for things like this SCOBY when making kombucha. So what happens basically is this biofilm of prokaryotes or bacteria kind of cuts off, creates a film on the top of the tea. So this is basically brewed tea. Creates a film on the top of the brewed tea that cuts off the exchange with the atmosphere. So it creates an anaerobic environment, which causes some of the components of its biofilm, yeast, which is not a prokaryote, but yeast to switch over from aerobic respiration to anaerobic respiration to start breaking down some of the sugars that are in here and the carbohydrates that are in here to start producing kombucha, right? So biofilms are very common. Prokaryotes will often live together in community like this. While metabolic diversity is one of the key components of being a prokaryote, many different types of prokaryotes carrying out and getting energy and getting carbon from many different sources, that's not the whole story, right? So there's huge diversity of prokaryotes that evolved over time through this process of rapid reproduction, frequent mutations, and a huge diversity of environmental niches, right? Different types of environments for them to live in, different food sources, different energy and carbon sources, led to the diversity of prokaryotes that we have today, which also laid the groundwork for eukaryotes to come about, which is what we'll talk about next.